Welcome to MLT Topics channel. Today we will be discussing about Introduction to Arbovirus. In this video, there will be brief explanation about classification, families of arboviruses, its general properties and laboratory diagnosis of arboviruses. Arboviruses, arthropod-borne viruses, are RNA viruses that are transmitted by blood-sucking arthropods from one vertebrate host to another. Viruses multiply in arthropods without producing disease. A similar ecological group is rodent-borne, robo-viruses, which are maintained in nature by direct transmission between rodents, and sometimes infecting humans by direct contact without the agency of arthropod vectors. Classifications. Arboviruses were sometimes named according to the disease they are associated with, such as yellow fever, chikungunya, or after the name of place of isolation of the virus, such as Kyasanur forest disease. Some were named after the geographical area. Example: West Nile fever and St. Louis encephalitis. Families of arboviruses are Tocaviridae, Flaviviridae, Bunyaviridae, Rheoviridae, Rhabdoviridae, and Orthomyxoviridae. Here is the list of diseases caused by arboviruses. General properties. Arboviruses share some common biological properties, like all members produce fatal encephalitis in suckling mice after intracerebral inoculation. They possess hemagglutinin and agglutinate erythrocytes of goose or a day-old chicks. Mosquito-borne arboviruses multiply in Aedes and Culex mosquitoes, while tick-borne arboviruses multiply in lizardid ticks. They can be grown in tissue cultures of primary cells, like chick embryo fibroblast, or continuous cell lines like vero, and in culture of appropriate insect tissues. They may also be isolated in the yolk sac or chorioallantoic membrane of chick embryo. In general, arboviruses are readily inactivated at room temperature, and by bile salts, ether, and other lipid solvents. Antigenic structure. 3 antigens are important in serological studies, namely hemagglutinins, complement-fixing and neutralizing antigens. Considerable antigenic cross-reactions occur among arboviruses. Here is the list of properties of different arboviruses. Pathogenesis. The virus enters the body through the bite of the insect vector. It multiplies in the reticuloendothelial system, and leads to viremia. In some cases, the virus is transported to the target organs, such as the central nervous system in encephalitis, the capillary endothelium in hemorrhagic fevers, and liver in yellow fever. Virus enters through mosquito bite. Multiples in reticuloendothelial system causing viremia. Transported to target organs which causes disease. Arboviruses cause febrile illness, with or without rash, and arthralgia, encephalitis, hemorrhagic fever, and characteristic systemic disease, yellow fever. All arbovirus infections occur with varying degree of severity, subclinical infections being common. Laboratory diagnosis. Diagnosis may be established by virus isolation or serology. Specimens collected. Blood, CSF, and brain may be used for isolation of virus. As all arboviruses are viremic, blood is collected during the acute phase of the disease. CSF is useful in encephalitis cases, but the best specimen is the brain. Virus isolation. Specimens are inoculated intracerebrally into suckling mice. The animal develops fatal encephalitis. This is the most sensitive method for the isolation of arboviruses. Tissue culture. Some viruses may also be isolated in tissue culture. Vero, BHK21 and mosquito cell lines are inoculated with specimens. Growth of virus in cell cultures is identified by immunofluorescence, hemagglutinin inhibition, complement fixation, ELISA, or neutralization tests. Virus isolation from insect vectors and reservoir animal. Such isolations are important to know the identification of arbovirus activity in the area. Serology. Using ELISA, serotype-specific IgM antibody may be detected in patient serum within 1 to 3 days after the onset of illness. The detection of a 4-fold rise, or more antibody titer by ELISA, complement fixation, hemagglutination inhibition, or neutralization test also provide a good evidence of infection.